This presentation will describe the development of a lymphoid panel based on next-generation DNA and RNA sequencing for clinical use. There are many unique features of the lymphoid panel for next-generation sequencing that we have designed. First, it is a 24-hour workflow. Our assay takes 8 hours to prepare, going from sample to DNA sequencing library. It is optimized for a 16-hour sequencing run on the Illumina machines, and the software takes about 15 minutes to run. The panel sequences both DNA and RNA and is compatible with both formalin-fixed cases as well as frozen cases. The software provides a number of selectable features which allows users to select what they want to see, including what genes they want to see, up-to-date features like new DLBCL groups reported by several high-impact publications, and the format of the reporting itself, which is based on current CAP and ASCO guidelines. The panel is initially going to be implemented as a lab-developed test for cell of origin MYC and BCL2 translocations. This slide summarizes how the whole thing works. So what we have is an assay that is available as a clinical grade reagent kit for purchase. The reagent kit allows the sample to be processed into a sequencing library within 8 hours. That library is sequenced on an Illumina sequencer using a 16-hour run, and our software connects to the Illumina sequencer and provides the results. These would be then charged to the patient's insurance as a targeted panel, and it is priced in a way that is immediately profitable for the hospital. The report includes cell of origin, MYC and BCL2 translocations, which can be immediately incorporated into the workflow as part of the standard diagnosis. In addition, the report provides information on targeted mutations with rich annotation, as you will see. This slide shows an example of a report generated by our software. Our software connects to the sequencer and automatically analyzes the DNA and RNA sequencing reads to provide this report. The report is divided based on expression, which is based on RNA sequencing, and measures a number of markers that are important in cell of origin, as well as EBV positivity and so on. And these are calibrated across a large reference data set from our paper in Cell and other publicly available sources. The second part has a genetic assessment, which includes assessment of BCL2, MYC, and BCL6 translocations, as well as a genetic assessment of actionable variants. This is annotated using current CAP and ASCO guidelines and points to targeted therapies or clinical trials where these are available. The genetic assessment also includes assessing reported genetic subgroups by Lou Stout, Margaret Shipp, and other groups that are available for the users to select as customizable reports. The software then provides an interpretation. In this case, this patient happens to have a GCB-type diffuse large cell lymphoma, which the software automatically assesses from their DNA and RNA sequencing reads. And finally, the software plugs in all the mutations and expression data and compares this to a large reference data set for over 1,000 patients that we published in Cell, and plots a survival curve that allows clinicians and pathologists to understand this patient's risk based on all these measurements. And I'd like to point out that something like this just cannot be done currently because the number of measurements is just way too many for any single method to easily process. This slide summarizes our implementation plan. We plan to implement this as a lab-developed test starting in March of 2019. The plan is to institute adoption at 5 to 10 different institutions that will perform the same validation using shared reagents and protocols. Each of these institutions need access to an Illumina sequencer and would plan to enroll all eligible patients for at least 6 months. And the patient's insurance would be billed for this test, and the test is priced at a level that would be immediately profitable for the hospital. We also plan to publish the experience with this panel as a separate publication, And Blue Cross and Blue Shield has signaled that they will likely participate as an observer and share data to establish cost and clinical effectiveness. Please feel free to email me or contact me if you have any questions about this panel or want to participate. Thanks for your attention.